For the introduction. Thank you for inviting me here to talk to you guys today. This is a talk that's based on a report that recently put out called Editing Biosecurity. So there are executive summaries here for your interest. And this
send it.
And this, I just want to make clear from the beginning, this was a kind of a group project, so I was part of a team. I had a colleague also at George Mason University, and then two colleagues from Stanford University who have backgrounds in bioengineering and infectious disease. So this was a multidisciplinary research project that we put together. And the team was supported by a group of a multidisciplinary group of experts with expertise in the life sciences, science policy, biosecurity, ethics, in order to provide as many perspectives as we could on both genome editing as a technology and also some of the risks of misuse that are inherent in the technology. We want to make sure we had enough different perspectives to be able to cover kind of a pretty wide waterfront. In addition to the
where's the full report? On the web?
That's right here. So in addition to the full report, we also produced 5 issue briefs and 2 working papers that provide much more detail about different aspects of genome editing and biosecurity. All of those issue briefs and working papers and the final report are on our website at editingbiosecurity.org. So if you're interested in kind of learning further details about any of these topics, I would definitely refer you to our site for that. So the genesis of this project was twofold. The first was that starting in late 2012, early 2013, there was a breakthrough in the field of genome editing called CRISPR that really revolutionized the ability to modify genomes in a very simple and precise way. And this breakthrough was something called CRISPR, which stands for Clusters of Regular Interspaced Short Palindromic Repeats. If you think the Pentagon's good at acronyms, biologists actually are masters at it as well. And CRISPR is based actually on the bacterial defense system, where the bacteria would recognize viruses that are trying to attack it. They would cut a piece out of the virus, insert it into their own genome, and then develop nucleases, basically molecular scissors, that could then attack the viruses if they saw that virus again with the same DNA. And the breakthrough was in 2012 and 2013, Jennifer Doudna, Emmanuel Champetier, and Feng Zhang at the Broad Institute figured out how do you repurpose this DNA recognition system and these molecular scissors, how do you repurpose them not only to work in bacteria, to work in all those types of cells as well. And so they were able to show that you can use this combination of CRISPR to precisely identify DNA in bacteria, in animals, including mammals like ourselves, plants, insects, fish. Every organism they've tried to use CRISPR on has worked. And so this provided this powerful new tool to identify precise segments of DNA and what is called the Cas9 protein, able to cut the DNA in a very precise way. And so this is a whole new capability that was started in 2012, 2013. By 2015, it had been recognized by Science magazine as the breakthrough of the year, so getting a lot of attention, not just among a small number of people who did functional genomics and biomedical research, but really a much broader appreciation of the power of this technology. About a month later, the Director of National Intelligence talked about genome editing in his unclassified worldwide threat analysis to Congress. And he talked about genome editing as if it was a direct threat to U.S. national security. And he said, quote, given the broad distribution, low cost, and accelerated pace of development of this dual-use technology, its deliberate or unintentional misuse might lead to far-reaching economic and national security implications. Moreover, he put that discussion of CRISPR in the section on weapons of mass destruction, right after North Korea's nuclear weapons, Syria's chemical weapons, and what Iran was doing. And so they were making a very clear implicit connection between this technology and its utility for developing weapons of mass destruction. And so this kind of raised our interest to understand better to what extent is this technology really a threat? What is the risks of misuse? And more importantly, how do we devise new governance mechanisms for trying to mitigate those risks at the same time sustaining the innovation you need to basically be able to harness the breakthroughs that we're seeing in the lab that have medical and public health and agricultural applications. And so that was kind of the genesis of our project. And what we're trying to do was do a more kind of balanced assessment of both the benefits and the risks of this technology, and look at to what extent is our current policy and regulatory system capable of dealing with these this new technology, and what other governance options are needed to try and fill the gaps or extend the reach of policies in ways that would prevent misuse. And when we talk about misuse, it's not just the traditional kind of bioweapons ideas of misuse. We're looking at the range of accidental misuse, reckless applications, and deliberate misuse by state and non-state actors. So we're trying to define the range of possible scenarios kind of broadly. We didn't want to kind of pigeonhole and just look at one particular set of problems. And at the time, there were a number of other projects that were also looking at genome editing. But ours is unique in the sense that it was multidisciplinary, right? By design, we purposely sought out people with expertise in different aspects of the life sciences, social sciences, the policy world, national security. And in addition, it's based entirely on unclassified open source materials. There are a lot of other reports that have been done for the government that are based on classified or restricted access types of things. And this was entirely an academic exercise, so our kind of charge and our approach was determined by our by our group. We were not being directed by a government agency or another kind of body. So we had a lot more kind of flexibility and freedom to look at what we thought was important. And in the study, we looked at genome editing on a couple of different levels. So it's important to understand that genome editing as a tool, and there are certain kind of technical characteristics that are relevant that I'll talk a little bit about. But it's not just about the tool. There's an entire process that's used to use these tools to make the genetic modifications that are desired. And these enable a range of capabilities beyond just cutting DNA. Cutting the DNA is just the first nucleus step. There are then multiple things you can do with the modifications to the DNA that we'll talk about. And finally, there's looking at genome editing as a field, looking at the whole ecosystem of the economics, the legal aspects, the kind of the social and organizational aspects that define who are the actors, who are the stakeholders, how are these things being developed and applied, and therefore who are the actors you need to engage if you want to engage in more governance or regulation or policy, or who are the people potentially affected. So we kind of use these kind of levels of analysis as a way of looking at the different aspects of this technology. So just by way of background, in the field of biology, the central dogma, right, is that DNA leads to RNA, which generates amino acid change, which provides the proteins and the enzymes and all the other biological molecules that are essential to everything that we do, right? This is life itself. And so the key takeaway from this is that if you have a greater ability to control DNA, you have the ability to control everything that flows downstream from that. And this is why the genome editing tools, especially CRISPR, has been so revolutionary, because it's given a much higher degree of control over DNA than previous techniques used. So I'm going to show you a short video that hopefully will illustrate this in a way that I could never hope to explain adequately. So what you see here is the this is the what's called the Cas9 protein. This large object is the protein that will actually do the cutting of the DNA. There is a guide RNA embedded in the Cas9 that will seek out the exact match of the DNA. Guide the Cas9 to the DNA. Will, as you'll see, it cuts the DNA, both strands of the DNA, and makes a complete cut of the site that it is. All technology, this worked 5 minutes ago. Okay. Right, so the guide RNA has latched onto the DNA that it was designed specifically to find, and the Cas9 protein will do these two very precise cuts. And then DNA will try and repair itself, and usually that process doesn't go very well, and so it will create errors and mutations. And usually what that means is that the resulting DNA will be a non-functional gene, so you can kind of knock out the gene by doing that. Or a next step would be to insert your own donor DNA so that you are inserting a new strand of DNA that could code maybe for a new gene or a different version of the gene. And so this would be a knock-in effect where you're adding new DNA, which could be an entire gene into the organism. And really the amazing thing about this is, right, this is a natural process that Mother Nature invented over a long period of time and perfected, and that we are now kind of repurposing and leveraging for our own purposes in ways that were not originally intended. Biologists hate this analogy, but I'm from an international security background, so I think about CRISPR as a precision-guided munition. Whereas the old-fashioned genetic engineering was more about carpet bombing and kind of brute force methods to make genetic modifications that could sometimes be random and incomplete. Here you have the ability to create a guidance package that will directly deliver your very precise munition to an exact target and do have tailored effects. And it could be either knocking out a gene, it could be inserting new DNA, or you could also use different Cas proteins that have been developed in order to modulate the expression of that gene. And so you don't actually change the DNA at all, but you're enabling it to either activate or interfere with the expression of that particular gene. So it's very customizable, can be very tailored to a wide range of different applications. And so in fact, the analogy that scientists do like is comparing CRISPR to a Swiss Army knife, because it is a platform technology that allows you to do multiple things with the same basic Cas9 protein. You just swap out this very small guide RNA to determine where it's going to go in the DNA, and you add on maybe one or two other payloads on top of that. It provides a lot of flexibility and a lot of versatility at a technical kind of biomedical basis. But what that also means is that it gives you the ability to make these genetic modifications not just in single-celled organisms, but in plants, animals, you name it. So again, very versatile in terms of its ability to be applied across the board. But genome editing with CRISPR is not just about the nucleases and the actual kind of proteins involved. It's not a single discrete activity. It is part of a process. And if you look at this process, you understand a little bit more what it takes to engage in a genome editing activity. And aside from the actual kind of CRISPR tool and reagents, there's a very strong bioinformatics component to this, because you need to design your guide RNA to target the DNA very precisely, and you want to make sure you're targeting the right DNA to have the effect that you want to have on the organism. So there's a lot of bioinformatics and software that goes into how do you predict and design the activity. And at the same time, there are a number of skills that you need to have in order to execute this properly. Part of it is bioinformatics. There is the need for having the ability to work in cell culture, because this is where you're cultivating the cells that you're editing. And then also skills, kind of basic molecular biology skills like molecular cloning are kind of the backbone of what people are actually doing in the lab. And the key thing here is that even though you do need a range of different types of skills to use this technology appropriately, this particular form of gene editing is kind of one of the more user-friendly versions out there. The previous tools that were used for genome editing relied on very sophisticated protein engineering to develop the tools to do these kinds of editing. And the number of people who have that protein engineering expertise is relatively small. But everyone in college-level molecular biology programs has taught how to do molecular cloning. So when CRISPR came out, as we'll see in a little bit, when CRISPR came out, there's already a huge latent user base that were able to adopt the technology very readily into their typical laboratory routines. And this is part of the reason why CRISPR took off so suddenly and dramatically. This process also illustrates some of the obstacles that we've seen arise in the area of genome editing and actually having these edits do what we want them to do. So the first step is just predicting what you need to edit, and we've seen that there are challenges in terms of being able to predict exactly how do we design the right guide RNA to target the DNA that we want, what is the DNA that we actually want to target in order to have the desired effect. There's a lot of kind of basic research knowledge that you need to have in order to figure that out. You also need to pick the right vector to deliver your genome editing payload into the target, and it depends what kind of organ or tissue that you're targeting. Different vectors have different strengths and weaknesses, so there's an expertise that goes along with how do you optimize the combination of the payload and the delivery system. And then one of the other major challenges, which you guys have probably heard of, is what's called off-target challenges. I said that CRISPR is a precision-guided munition, but even these things go astray sometimes, and they will do collateral damage, and they will make edits in parts of the genome or in other cells that you did not want to happen. And so this is kind of a recurring problem. And especially when we talk about kind of medical applications and clinical applications, this is the major obstacle, because you cannot, the FDA cannot approve a drug based on genome editing if they say, well, it'll 10% of the genes that modified will be the wrong genes, right? And that causes major problems from a regulatory perspective. So, and this is an area that's receiving a lot of attention. How do you minimize these off-target effects and try and restrict the impact of CRISPR, the genome editing tool, to the specific strain of sequence of DNA that you're interested in? There's also the problem of mosaicism, where if you see at step 5, some cells will be properly edited, others will be maybe partially edited, and others will be totally unaffected. And so this is still not a 100% complete process. It's still stochastic, and still there's some percentage of genes that will be edited properly, and others will not. So how do you increase the yield and the completeness of your gene editing experience? And again, this is an area that's getting a lot of attention. And so, so there's early on, recognized huge potential for this technology, not just in the lab, but in the application space. Another just very important thing to highlight is the fundamental importance of having a very strong understanding of how genotype maps to phenotype. How changes to an organism's DNA affects their behavior or their attributes. And this is this area of functional genomics that is a huge area of study to understand how do changes in an organism's DNA actually affect their behavior and their performance and their attributes. And what this highlights is the importance of understanding the language that you're editing. So even if you have the world's best editing tool, right? Even if CRISPR was like editing a Word document, if you don't speak the language, right? You're making these edits that are very clear and precise, but you have no idea what you're actually saying. And what you're editing might actually wind up being unintelligible, or you might have very unanticipated side effects. So even if I misspell the word genome, right? I'm suddenly doing research on gnomes, not genomes. Same thing, if you do not understand exactly what you're editing and why you're editing it in a genome, you could have unanticipated effects as well. So even though the tool itself is relatively easy to use and it uses a skill that's very common in lots of laboratories, it still requires a very deep fundamental knowledge of the genome of the organism that you're editing, right? So just because you're an English major doesn't mean you can go read Sanskrit, right? And same thing, just because you are a virologist does not mean you can then go off and edit a bacteria. You can edit mammalian cells. So there's still a very deep level of knowledge that's required. So there's still a lot of expertise that goes behind this, but it gives those who have that expertise a much easier tool to use to make these kinds of these changes. Now, I talked about all kind of the things that CRISPR does well, and so some people have talked about kind of the CRISPR craze, and this is sensationalized. There's all this hype. There's lots of magazine covers and internet stories about all these magical things that CRISPR is going to be able to do, and there's something to that. But at the same time, there really is something different about CRISPR that has explained why it has become so widely adopted in so many lives for so many different applications. Again, from public health to human medical applications, agriculture, biomanufacturing, across the board. And what you see is that there are some biological technical advantages that this tool has that the biologists really like, and explains partly why they're able to use it so widely. But at the same time, there are advantages to accessibility, scalability, economics of this, and just the ability to be able to, the versatility of this tool to be able to use across so many applications. In addition to kind of the technical side, there are some organizational legal issues here as well. So there's actually, so that you guys might know there's a big patent fight between Berkeley and Harvard, MIT over who owns CRISPR and who owns the patents. Even though that is a big fight, the intellectual property environment for researchers is very permissive. If you're just doing research, it's very easy to get all of the CRISPR reagents and items you need. It's only if you want to commercialize that you then have to fight with these big institutions about who owns the patents. So scientists have very easily been able to order whatever CRISPR plasmids or reagents they want and use them in their research. And there is a group called AGENE, which is a nonprofit group based in Cambridge, whose whole purpose is to facilitate the distribution and use of tools like CRISPR and plasmids. And you can see this based on some of the numbers, just how popular CRISPR has become in terms of their distribution and just how global that's become as well. The latest numbers I have is that AGENE has distributed more than 140,000 plasmids in the last 6 years to over 78 different countries. At the same time, companies and labs that use this technology will then upload or deposit their new plasmids with AGENE as well. And so it becomes, again, this cycle where you now have the new latest and greatest plasmids and CRISPR payloads at AGENE, and then someone else can order them, and then when they modify that and they make it better, they add that back to AGENE. And so it becomes, again, this kind of virtual cycle of being able to, scientists being able to leverage off one another in a very easy way, where AGENE has become this centralized repository and brokerage service that kind of takes a lot of the logistics and the hassle of scientists mailing this stuff back and forth to each other. You have this one organization in Cambridge that does it for everybody. So it's much more efficient. And as a result, they charge only $65 per CRISPR plasmid. Other genome editing tools cost hundreds of dollars if you want to use it. So this enables more labs to get access to it, and then the labs that are very well endowed can run large, huge parallel programs with lots of different CRISPR plasmids at the same time. So it's both kind of an access issue and it's a scalability issue.
Can I ask you just a
Yes.
quick question to interrupt you? Can anyone order it from them, or there are some certain regulations, restrictions?
All right, so anyone can order from AGENE. They have an online form that you fill out. So as long as you have a credit card that's valid and you have an address that they can actually ship to, then you can order it. Right, and again, like they ship to 78 different countries, so they don't have any restrictions on where they send their plasmids to.
And what do you actually receive? A little tube with the, what is the product?
Right, so it would usually be a tube with your CRISPR plasmid, and they have different forms that you can receive it in depending on what your needs are. They also sell CRISPR kits, which would be a larger package that would have not just the plasmid in it, but other reagents, other equipment that you need to run your experiment. Again, depending on what you're doing, there are different kits that you can use at different stages. So they also sell the vectors, and so you could get a kit that would have the vector and the CRISPR payload, and then you would yourself, combine the two in your lab and insert it, inject it into whatever cells you're working on. But there are lots of different ways that you can order it. And so again, it's very customizable and it's very much tailored to the individual scientist and what they need, and it lets you mix and match. So again, it's that accessibility that they make it very easy to engage in this kind of research and order these materials that you need. And there are companies that do this as well, but AGENE as a nonprofit is kind of the leader in this. But some of the more specialized things, you probably need to go to a private company to get. Okay. And so because of all this, right?
Okay, just one little question. What's the level of education of somebody to be skillful to deal with it? BA or even less than BA?
It depends what you're doing. So there are community labs that run CRISPR classes where you come in literally off the street, and they have already bought a kit, and they will have yeast or E. coli that they will have the kit that you will then use to physically combine the plasmid into the vector, inject it into the yeast, and then see if you can make it glow green or change color or something like that. So at a very basic level, you just need very minimal kind of lab skills and knowledge, because these kits kind of have, they black box the process. You don't have to understand what is happening on a molecular level. You just have to understand, okay, here's the 10 steps you need to follow to do it, right? But that's very limited, right? These kits let you do very limited things, like insert a green fluorescent gene into E. coli. That's all they can do. If you want to do something more sophisticated, like edit a mammalian cell, you want to edit a human embryo, right? That would require a much higher level of skill and expertise and resources. And this is part of what makes CRISPR so versatile, is that there are very basic tasks you can do it with very minimal training, but at the same time, if you have that level of expertise and knowledge, right, you can do much more sophisticated things with it as well. And so this is part of the reason why you see this in everything from the Broad Institute and Harvard, MIT, down into community labs, where people are paying $150 to go in and take a class about how do you CRISPR yeast or how do you CRISPR E. coli. And so that's part of the nature of this technology, which is unusual. Right? Most of the genetic engineering technology is restricted to traditional biomedical research facilities or universities, but CRISPR has that potential crossover into a much broader use. Again, very limited applications there, but still you can actually engage in gene editing experiments, right? If you have a couple $100 in a free weekend, and so several of my students have done that, and so it's and they have some technical background, but but it's not something that requires you to be a Ph.D. molecular biologist to do. So as a result of all this, right? Like I said, there are a number of fields of where genome editing is being applied for human health, agriculture, biomedical research, industrial biotechnology, for just an incredible diverse range of applications. And as a result, lots of different types of stakeholders are working in this technology that is, again, relatively new. Because there are lots of potential commercial applications, you see that the private sector playing an increasingly large role in this space. And there's lots of the patent fight. There are lots of companies have already been spin off from the main labs at Broad and at Berkeley to try and commercialize this technology for different applications. And what's going on in the genome editing space is not specific genome editing. It's part of this broader trend we see where private funding of life sciences research is becoming increasingly the norm instead of the exception. So in the last couple of years, the private sector has emerged as the largest funder of life sciences R&D, more than the government, which is a dramatic change. Also, I think last year for the first time ever, there are as many science and engineering PhDs working in the private sector as there were in academia. So increasingly, we're seeing the locus of innovation in biotech in general, but CRISPR specifically, shifting into the private sector, which means that there are new actors who are working with this technology that don't have necessarily the traditional awareness of some of the kind of safety and security issues I'm going to talk about in a minute. And there's a growing awareness that even though there are billions of dollars at stake here, and there are predictions that genome editing will be a $10 billion business in the next few years, there's a small but growing realization that all of that growth and innovation could be undone by a biosafety breach, by a biosecurity incident, by some reckless application of the technology could derail the progress that's being made. And so part of what we're trying to do also is educate the folks in the genome editing world to, okay, here are the kind of potential risks that you should be thinking about, because even if you're not going to do it, even though your lab is not going to do it, if this happens in some other lab or even another country, it could have an impact on the entire field. And so you as a community need to be taking these things seriously, proactively, in order to prevent some of the worst case kind of reactions down the line. So now to provide you some of the kind of background on what CRISPR is and what genome editing is and how it works, let me flip it over a bit and talk about how genome editing can be misused. And in the study, we kind of identified 4 categories of misuse. Kind of traditional kind of biosafety concerns about developing new and improved biological weapons, biosafety, accidental releases of a gene-edited organism into the wild, reckless applications, of either unapproved or unproven products being sold to the public, and the issue of dual-use research. And so let me talk about a little bit on each of these. So in the context of dual-use research, CRISPR is really dramatically accelerating, expanding our understanding of that functional genomics, right? How genotype maps to phenotype, which is kind of the holy grail of functional genomics. And at the same time, it's giving us more tools to exploit the discoveries that we are making as well. Again, because it's this versatile Swiss Army knife-like platform, it's both a discovery tool and an exploitation tool. And unfortunately, that means that these knowledge can be used for good or for ill. Right? This is the classic dual-use dilemma that we see. Genome editing is the most latest manifestation. We saw this with synthetic biology in the 2000s. This was the original concern about recombinant DNA in the 1970s. So this is not a new concern, but this is definitely a key issue for genome editing. One of the problems that we're facing now in this dual-use research space is that there's a very small segment of the life sciences community that actually is aware of, familiar with, cares about dual-use research issues. And those are scientists who work with a certain class of dangerous pathogens called select agents. And these are pathogens that are regulated by the US government and to prevent biosecurity incidents from happening. And only scientists who work with that smallest, about a dozen pathogens, are regulated in terms of what kind of experiments they can do with these pathogens. If you work with a bacteria that is non-pathogenic, even if you are trying to make it pathogenic or make it dangerous, that is actually not covered by our current dual-use regulations. So there's a very small cluster of people who have to worry about this. If you work in immunology, cancer therapy, gene therapy, you are not subject at all to any kind of dual-use research oversight. And there's no one there looking over your shoulder to say, hey, that experiment you're doing with this oncogene, and you're putting it into an inheritable vector, and you're inducing human-type cancer in a mouse model, right, for research purposes. Gee, that could be potentially dangerous. And if you use that vector and you gave that to a human, right, you would be inducing, in theory, you could use that vector to induce cancer in a human being. Right? There's no one looking over the shoulders of these people saying, what are you doing and why are you doing it and how do we do this more safely, more responsibly. So this is one of there are whole groups of stakeholders that are working with this technology with lots of dual-use applications that have any kind of government oversight and even kind of awareness, education, that just they don't have that awareness at all. The second part of this, like I said, this technology is increasingly being developed in the private sector. The private sector is totally exempt from these kind of dual-use research oversight that the US government has. It only applies now to federally funded research. So if you're a private company and you're using money from the Gates Foundation or from stock or you do not have any obligation to check that your work has any kind of dual-use research implications and check them on the U.S. government. And we've seen cases where people, were doing, research that does raise dual-use research questions and it could be misapplied, but because it was a totally private enterprise, they had no obligation to actually, check their work or have anyone give them advice about what they're doing and how to kind of mitigate some of the risks.
Is there work on select agents in that milieu, or does it only happen with federal funding and oversight?
So only federally funded research for select agents is subject to the dual-use research.
But there is select agent research in the private sector, is what you're saying. It just doesn't get oversight. Is that right?
It wouldn't have oversight for dual-use research purposes. It would still have oversight in terms of making sure it's stored safely and securely.
It's like
but if you're but it doesn't have dual-use oversight. So and like I said, the private sector is increasingly a major player in this space. So there's increasingly large and growing loophole in our dual-use research oversight where the private enterprises are driving this research and they're just not aware of and have no obligation to worry about these kinds of issues. CRISPR has also enabled the creation of some entirely new types of genetically edited organisms. The most famous one is a gene drive, which enables you to, quote unquote, drive a gene through successive generations of a species, even if that gene does not provide any fitness advantage, right? So it kind of takes Mendelian genetics and survival of the fittest put to the side, and you're able to, by splicing the CRISPR directly into the genome, that CRISPR will replicate itself in every chromosome that it encounters during the reproduction of your edited species. And so instead of it just being a 50% chance of being inherited, right, you have 100% inheritance of that specific gene in the edited organisms. And so this provides a way to drive a gene into a population, at a species level. And so again, a very powerful tool that's being used to explore how do we, for example, reduce the ability of mosquitoes to infect people with malaria. How can we use this to maybe, kind of sterilize and therefore wipe out certain agricultural pest species that are causing problems. The one kind of caveat to this is that these kind of gene drives only work in species that reproduce sexually, that have very short reproduction times on the order of 1 or 2 years, and this means that this is not at all feasible to use in human beings, right? Or it just takes too long for us to reproduce for this to have any really meaningful effect. But for insects and for rodents, right, these are the types of animals that are being developed in labs that could pass a gene drive through that species, relatively quickly and have a major impact on the population and the broader ecosystem as well. And so a lot of concerns from a safety perspective of if one of these gene drive organisms gets out of the lab, what effect it would have on the ecosystem in the wild. And right now, the biosafety regulations in the US do not cover this at all. All our biosafety regulations revolve around pathogenic organisms. These fall outside the scope of that. There is a group of scientists that have proposed voluntary guidelines that would put in place certain protections so that if you're working on a gene-edited mosquito, you could only do it in an environment where that mosquito would die if it escaped the lab, and you'd have multiple levels of containment and biocontainment, but those are totally voluntary, and we know that there are labs that are not following those guidelines, and that they're doing work with gene-edited organisms in a context where they're not following all of the kind of best practices from a biosafety perspective. So there's a big gap in our biosafety regulations that this falls through right now. Another class of concerns we talked about were regulatory loopholes and lax enforcement of regulations that would allow unscrupulous researchers or companies to sell unproven and unapproved products to the public. During our research, we purposely did not discuss the issue of human germline editing, but we released our report literally 1 week after He Jingqiao announced at an international summit on human germline editing that he had edited the embryos that resulted in the birth of 2 live babies in China, Lulu and Nana, who had their genes modified to confer resistance to HIV infection. And this was a huge bombshell that he dropped at this international summit because the genetic changes he introduced into these embryos that grew into these two babies will be passed on to future generations. Right? This is what germline editing is all about, putting in permanent genetic modifications that will be inherited by future generations. And this was condemned by the summit. He's now subject to investigations in China. His American collaborators are also being investigated by their different universities, but this has still not been resolved. But this kind of validated our concern that there's this potential for reckless application. It's not, he wasn't doing this kind of maliciously. This wasn't an accident. He did this purposely, but he did it in a way that was unethical, and it was scientifically kind of sloppy as well, and has unforeseen effects on the two babies that have been born, and clearly is having a huge impact on the field of genome editing in general, and has really set back the idea that you can use this technology for therapeutic purposes.
When you say unethically, you mean secretly?
It was done secretly. There's a question of whether he really got real informed consent from the parents or not, because the way he described the experiment in his paperwork was not what he really did. He purposely was targeting HIV-positive parents and promising them that he'll make their children resistant to HIV, there's a huge social stigma in China about this virus and this disease. And so there were on many levels he was not engaging in ethical behavior with both internally and with the patients. And then the way he also discussed this, the way the international community found out about this was he posted YouTube videos bragging about what he had done, and I think he was even trying to pitch one of his companies as saying, well, if you too want your child to be, resistant to HIV, come to our company. So lots of very dubious ethical issues, and he turns out he'd actually been talking to several American scientists and ethicists about this, and most of them had told him, you shouldn't be doing this, but it's not entirely clear if he made it clear to them that he was actually doing it, or he was saying, well, hypothetically, if I did this, what would you say about this? So that's still being kind of investigated, but there is some evidence that there's some people who definitely knew what was going on, and they didn't kind of raise any red flags, they didn't ring any alarm bells, they didn't tell anybody about what was going on. So there's some real concerns that the scientific community, which has been focused on self-governance and how do we take care of our own and avoid excessive government regulations in exchange for scientists policing themselves, that system seems to have failed here, right? Because there were scientists who did know what was going on and they didn't say anything until he went onto YouTube and made this.
And who is doing the investigation?
Within China, I think it's the Ministry of Science. I think his university where he was technically on leave from, his university is investigating it. And then there were people he talked to in the US at Stanford and at Rice University. And he actually had a direct scientific collaborator at Rice University. And so those universities are also looking into this. And there could very well be other inquiries as well, but there's a lot of attention. And it remains to be seen what will the consequences be. Right? Will he just get a slap on the wrist? Will he be scapegoated by the Chinese or by others for this? Will his collaborators and the folks who kind of should have known better, right? What will come out of that? We don't know. So when we talk about kind of the ethics of this and the norms around how this technology should be used, this is going to be a very important test case. And there are definitely people who want to downplay this incident and not let it tarnish the potential of genome editing and germline editing in general. But at the same time, right, if you have someone who's so blatantly violated all of these ethical and probably legal standards as well, right, if he does not kind of held to account, then that would just encourage others who say, I guess I too can gain fame and fortune doing this, and I'll ignore clinical trial, I'll ignore IRB, I'll ignore the ethics that are not enforced at all by governments or by institutions. So this is an important case to keep an eye on. And it just highlights, even though most of the research in the US is being done on somatic cell therapy, which means making changes to genes and that are not transmittable and are not inheritable to future generations, right? There's still lots of potential for even somatic gene therapy to be done in a way that's unproven or unapproved by regulatory authorities, but yet sold to the public, and people, engaging in what they think is medical treatment that actually is, either useless or possibly dangerous. So this is a major concern in general. And if you have enough cases of this happening, it's not only bad for the individual, but if this then causes the entire field to suffer, because now all the genome editing is seen as being unscrupulous or dangerous, right? That's a major problem as well. Okay, then there's the whole realm of kind of the direct kind of biosecurity concerns about how this technology can be misused to deliberately create biological threats. And so we took a close look at several areas, but in doing so, we also have to be mindful of what is called information hazards, right? That we don't want to disclose vulnerabilities that someone could then exploit. We don't want to necessarily give quote unquote bad guys new ideas about what they could do. But at the same time, we had to provide plausible, concrete, hypothetical scenarios and vignettes that could illustrate vulnerabilities both in the technology, but also gaps in our government system, and looking at how those gaps could be used by a nefarious actor to develop something that would be dangerous and harmful. And so part of what we did was we developed some scenarios around these areas, which I'll just talk about briefly. But we also were looking at a couple of areas that have been discussed already in the literature. So these we're not really creating anything new here, but looking at to what extent does genome editing really provide a value added here, or to what extent is this something that is not there's no real capability here. It's a new technique. It's cool. It's cheap, but it doesn't actually fundamentally change the threat landscape. So one of the things we looked at was the potential for modifying pathogens to become more dangerous, whether it's making more lethal, more transmissible, resistant to medical countermeasures, or what have you. And there's definitely a possibility for genome editing to be used in that context, but it was also clear that there were also a number of other genetic engineering tools that have been developed that are well optimized for working in different types of pathogens. and that you don't really need genome editing in order to make bacteria more antibiotic resistant, or to make a virus resistant to antivirals or circumvent vaccines. So genome editing is yet another concern, but it was not a game changer in that regard. What was more disturbing was the ways in which CRISPR enables new types of biological attack vectors that do not rely at all on pathogenic organisms. And this is a completely new area. So for example, we looked at the potential for weaponizing gene therapy, right? The same type of vector that's being used to treat cancer or treat genetic diseases, if you change out that payload from being a therapeutic one to being one with deleterious gene elements in it, could be used to either cause cancer or disrupt key physiological systems in humans as well. And on top of that, there's research being done on vectors that can cross the blood-brain barrier, which would be great for treating neurological disorders. And there's lots of work being done on vectors that are inhalable, right, which would be good for treating respiratory issues and genetic defects that are found in the lungs. But again, those same type of vectors obviously have clear dual-use applications that make them either easily aerosolizable, so for large-scale dissemination, and if you don't have a way to cross the blood-brain barrier easily, right, that opens up your neurological systems for attacks that were not previously possible. In terms of hijacking the microbiome, which is something else we looked at, right? We are composed of billions of also bacteria that have beneficial purposes, that live in symbiosis with our human cells, and that we found that this microbiome is tied into our endocrine system, our immunological system, our neurological system, and so there are now specific diseases and conditions that manifest themselves in in our immune system, but the source cause actually is in the microbiome. And so if you now have a way of editing the bacteria in the microbiome to either produce harmful biochemicals or suppress the production of things that you do need in your body, that would be a backdoor way of affecting these other physiological systems that are dependent on the microbiome as well, right? And this would be based on vectors and genetic elements that are not at all pathogenic, right? We're not talking about the typical list of traditional biowarfare agents that we worry about. So this would be a totally new way of causing harm.
Why don't we wait this time?
Okay.
Because I want to make sure we have
Sure.
time for Q&A.
Okay, so the one last big point I want to make here is that what this means is that in the cybersecurity world, they talk about attack surface, which is trying to measure the number of vulnerabilities, the severity of the vulnerabilities, how easily access these vulnerabilities are in a computer system. Genome editing has increased the attack surface for biosecurity. There are new types of vectors, there are new sources of biological hazards, there are much more diverse set of actors who can access these technologies. There are more populations that are susceptible to these hazards than we've seen before. And our current kind of biodefense paradigm, which is oriented around a specific list of named pathogenic agents, and we have defenses that are specific to each individual agent, right? So we have a vaccine against anthrax, only works against anthrax, doesn't work against anything else. That's most of our biodefense is very agent specific. By enabling the creation of attack vectors that do not rely on pathogenic organisms at all, that has potential to totally circumvent our entire biodefense system as it currently stands. So major potential disruption to the way that we are able to do biosecurity today. And again, because all this is coming out of the private sector, it means that there are stakeholders in the private sector who need to be engaged, and because this is research being done in labs that specialize in immunology, cancer therapy, gene therapy, there are lots of other stakeholders who fall outside of that traditional biosecurity domain who nonetheless need to be brought in order to figure out ways to how do we identify some of these risks and mitigate them, but do so in a way that does not stymie the research, does not get in the way of the innovation, because again, there are huge potential health benefits for this technology down the road. So how do we find that balance? And oops. Okay, well again, my last slide was supposed to be the editingbiosecurity.org website, so if you've got more interest in some of these issues, you can go in there and look at some of the details. But with that, thank you for your attention and welcome any of your questions about this.